All right, so this time around we're going to be doing our first of hopefully a weekly thing, which is a Journal Club Thursday. Maybe I'll come up with a better name for that. For now, that's how it's going to have to be. So the idea of this is either we're going to read a paper, basically introduction through at least the end of results, maybe discussion as well. And we're going to do that with the open access ones, and particularly our papers from this lab. But thinking about maybe doing a live journal club style kind of review of like the non-open access papers and papers of interest to the lab, particularly community in general. So if you're into that sort of thing, welcome. And why don't we get into this recent paper that we published in PLOS Genetics. It's a review on transgenic optical tools. And yeah, let's get into it. All right, so let's begin with the introduction. Biological inquiries utilizing live organisms offer superior relevancy to cell culture and fixed tissue studies in unraveling the expansive mysteries which remain in cell developmental and disease biology, because the research is performed in the most functionally relevant environment. A panoply of tools has been developed to interrogate these perplexities of nature, but none as exciting as the growing number of optical tools which have recently become available for use in live organism studies. As such, this review will focus on the optical tools that can be readily used as in transgenesis, and will not cover the tools for interrogating biological processes via fixed samples, non-optical means, or by using tools that require external biochemical treatments. In the first instance, there are several available extensive reviews covering advances in chromatin architectural characterization techniques, fluorescence in situ hybridization related methods, and optogenomics and proteomics. Similarly, improvements in multiomics, mass spectrometry, DAM-ID, and bio-ID have been recently chronicled. Exciting new in vivo dyed drug and other external treatment techniques, while beyond the scope of this inquiry, are exploding in utility in basic and translational molecular biology fields. Transgenic models recapitulate the natural setting of biological inquiries while reducing the frequency of required optimization steps and autonomously reproducing experimental toolkits. Here, we endeavor to compile a reference for anyone seeking to understand or develop new transgenic models expressing optical tools for visualization, quantification, and/or manipulation of subcellular and tissue-level molecular processes. All right, that brings us to Section 1, Advanced Tools for Visualizing Subcellular Functions in Vivo. Fluorescent transgenic protein detection tags is the first subsection. While GFP has only been used as a transgenic tool for less than 30 years, transgenic fluorescent markers for molecular biology have advanced to the point that mere labeling of proteins with even some of the most optimized fluorophores is sometimes regarded as banal, with growing criticisms of these fluorescent proteins' limitations. Maturation rates of fluorescent proteins have been a particularly popular target of recent optimization studies to ameliorate unavoidable delays in protein detection of target fluorescent protein fusions. However, such limitations are now being avoided altogether through an alternative approach whereby fluorescent proteins are ubiquitously expressed separately from the target protein, but directed to the target protein through tag binding in vivo. In this way, fluorescence can be detected immediately upon translation of the target protein. These secondary attachment fluorescence systems are generally termed tags, with one of the most popular transgenic tag systems being SunTag. The SunTag system makes use of two short peptides with high affinity to one another to recruit fluorescent proteins to target proteins. A fusion protein is constructed consisting of a protein of interest and up to 24 repeating sequences that allow for complementary attachment via a second fluorescent protein fusion protein. Specifically, a single chain fragment variable antibody is SCFV antibody is used, which recognizes GCN4. The GCN4 repeats fused to the protein of interest are used to recruit SCFV-bound fluorescent proteins to tag the protein of interest for detection. SunTag's scaffolding system produces increased brightness because several fluorescent molecules can simultaneously attach to the protein of interest. Llama tags are another protein tagging system, allowing for faster visualization in vivo due to their use of already mature fluorescent proteins. Llama tags use optimized nanobody fusion proteins to directly recruit fluorescent proteins to proteins of interest. This mechanism, and that's these here, B and C, this mechanism therefore requires creation of only one transgene to be used and allows for transgenic addition of the tags into fluorescent protein expressing animals, which are now ubiquitous. Okay, next subsection. Real-time transcriptional visualization. Rapid fluorescence detection via pooled mature fluorophores at the ready has grown in popularity not only for visualizing protein locations, but also for detecting gene expression and mRNA dynamics in vivo. By far, the most popular system for real-time transcription detection are the MS2 and PP7 systems, whereby MS2 coat protein, MCP, and PP7 coat protein, PCP, fuse to fluorophores, bind MS2 and PP7 RNA stem loops respectively. These have been used to generate a broad array of transgenic animals and computational tools to track gene expression timing in live imaging experiments. In addition to these popular mRNA visualization options, multiple other aptamer coat protein pairs are currently available for transgenic studies, including BGLG, U1AP, Lambda N22, HTLV1 Rex, TATREV, and several less tested variants. The two-part aptamer binding EIF4A provides an intriguing twist to the system, providing a reliable, straightforward method to label RNA without ubiquitous background fluorescence by fusing two parts of split fluorophores. Further, RNA binding Pumilio allows for sequence modification to bind to chosen engineered 8 nucleotide sequences of RNA and has been implemented in the PUMBI labeling system to label adjacent mRNA sites to utilize split fluorophores. The major limitation of most tag systems being the necessity for high background fluorescent signal from a pool of overexpressed fluorescent proteins poised for binding to the tag. These split fluorophore approaches present significant advancement by eliminating this excess fluorescent noise. Similarly, DNA sequences can be targeted transgenically for visualization using DCAS9 fluorescent protein fusions to target sequences in a guide RNA-directed manner, or via traditional engineering of zinc finger nucleus fluorescent protein fusions. All right, that brings us to Section 2, Transgenic Optical Sensors. Subsection 1, FRET-based sensors. Beyond simply labeling cellular components, a growing array of transgenic optical tools for measuring subcellular and tissue conditions have arisen in recent decades. The field of transgenic biosensors has, again, bounded forward from the earliest methods, such as fluorescent recovery after photobleaching, FRAP, in a mere few decades. These remarkable nano and microscale autobiological devices present a unique opportunity in the fields of developmental and disease biology, allowing for measurements where even the most modern and advanced exogenous technologies are insufficient. Perhaps the most versatile of these transgenic instruments is proximity detection via Forster resonance energy transfer, FRET. In this method, two proteins are labeled with fluorophores capable of fretting with one another, and the distance between them can be calculated when they're in sufficient proximity to one another. Mechanical forces are known to be significant in several developmental and disease pathways. A particularly innovative extension of this method is the creation of several tension sensors. Because the distance between two fretting fluorophores can be detected optically, fusing compatible fluorophores together, separated by a molecular spring, generally spectrin or flagelliform repeats, allows for the measurement of force on the fret pairs via a simple calculation of the measured distance and the known spring-like characteristics of the separator. These sensors have been developed in mechanical components of cells and tissues, ranging from extracellular matrix and cell-cell connections, cytoskeletal connections, and all the way into the nucleus and onto the genome itself. An innovative use of FRET in sensors has facilitated the detection of membrane voltage potentials via opsin-based ACE sensors. In the ACE sensor systems, a fluorescent protein is fused to an opsin with which it can fret. When the intracellular side of the membrane gains a net positive charge, increased fret reduces fluorescence of the fluorophore. ACE sensors now come in a variety of spectral flavors, including red, Varnum, and green, ACE2N, M-neon. Another creative use of FRET to sense subcellular conditions is FLUO. While optical sensing tools have long been a field of inquiry and optimization, FLUO cleverly uses a flavin-based fluorescent protein, FP, as donor, with a YFP acceptor to detect molecular oxygen within cells. YFP is sensitive to O2 levels, while FP is not, allowing for detection of higher fret efficiency in O2-rich cells. Caspase activity can further be measured using FRET pairs separated by caspase cleavage sites, thereby providing reduction in FRET as a readout for caspase activity. ATP concentration is an important subcellular condition which has motivated the creation of a multitude of sensors. The Transgenic A-team, I lost my place, I'm sorry. The Transgenic A-team sensors utilize modified ATP synthase, epsilon subunit, transversely terminally fused to a donor and acceptor fluorescent proteins, such that binding to ATP or magnesium ATP brings the fluorescent proteins into proximity, whereby increased FRET efficiency can be detected. Next subsection, bioluminescence-based sensors. A somewhat similar method for functional and proximity detection to the FRET-based sensors is the use of bioluminescence resonant energy transfer, BRET, whereby a bioluminescent molecule is used to shift the emission fluorescence of a fluorophore in sufficient proximity via molecular transference of the bioluminescent enzyme's emission to the fluorophore. FRET detection in FRET sensors, while precise, has proven difficult to effectively implement in vivo, as autofluorescence and relatively weak signal-to-noise, making alternatives like BRET attractive. However, while a panoply of BRET sensors exist, for example, calcium sensing with Lucid, CAMP sensing with Camiel, cytoskeletal tension sensing, O2 sensing, caspase activity sensing, and protein of interest interaction detection, current iterations remain shackled to the requirement of exogenous introduction of their cofactor substrates. As such, these tools remain distinct from pure transgenic optical tools. Despite this, recent advances have been made to achieve autoluminescence from bioluminescent systems by introducing genes that ultimately catalyze endogenous synthesis of chemical cofactors. Conversely, a clever employment of Aquaria victoria, apoaquarin, allows for calcium ion sensing without its bioluminescent cofactor requirement in the gap sensors. In these sensors, binding of calcium ions to aquarin shifts the excitation maximum of GFP, allowing for calcium ion concentration quantification via ratiometric fluorescence measurements. That brings us to the next subsection, modified and permutated fluorophore sensors. A methodological extension of the split fluorophore mechanism described above for in vivo tagging of RNA with low background is a class of sensors utilizing split fluorophores. In most of these systems, interactions of the proteins labeled with complementary subunits of the split fluorophores are detected by fluorescence via sufficient proximity of the subunits. While this method is similar to the FRET pair systems above, it has significant benefits and drawbacks in comparison. Split fluorophores confer a binary interaction measurement and not a distance, but offer higher detection resolution with less background, and require less sophisticated imaging and analysis techniques than FRET systems. Thus, split fluorophores have also been used to develop binary force sensors, where the subunits are separated by a flexible linker, allowing for subunit separation upon sufficient tension. Fluorophores have further been modified by rearrangement and insertion into their amino acid sequence to produce various effects termed circularly permutated fluorophores. A remarkable application of such circularly permutated fluorescent proteins is the strain sensor PRISM. Proximity imaging, PRIM-based strain sensor module PRISM facilitates detection of F-actin myosin 2 strain via ratiometric fluorescence following 490 or 390 nanometer laser activation. Strain on PRISM changes the orientation of tandem contacting GFP and circularly permutated GFP fluorescent proteins, making the optimal activation wavelength 390 nanometers and 490 nanometers detached from F-actin or under strain, respectively, as well as increasing fluorescence under strain. A further implementation of a circularly permutated GFP was used to construct a calcium ion sensor, GCAMP, which increases in fluorescence due to conformational changes induced by calcium ion binding. The reduced efficiency of circularly permutated GFP in the unbound state has the additional key advantage of increasing the signal-to-noise ratio of GCAMP relative to alternative strategies. The Percival sensor permits in vivo measurement of ATP-ADP ratios via measurement of fluorescence ratio from a circularly permutated monomeric Venus fused to GLNK1 when excited by 490 or 405 nanometer wavelength lasers. Further, use of modified fluorescent proteins to be sensitive to other molecules has also been accomplished, as in Clopensor, which uses a modified chloride ion sensitive GFP fused to DS-Red-M to detect chloride levels in cells by ratiometric fluorescence. All right, that brings us to section 3, optical tools for real-time subcellular manipulations. So subsection, activation inactivation systems. Optogenetics is a more recent technique that facilitates convenient manipulation of cellular function. With the use of light, proteins can be utilized to affect the function of the cell. Optogenetics makes use of light-sensitive proteins, either artificial or naturally occurring, and adjusts their functionality through adjusting their secondary, tertiary, or quaternary structure. There are a myriad of techniques that can be affected with optogenetic tools, but all are initiated by a chromophore or light-absorbing amino acid. Optogenetics is commonly preferred over chemically inducible systems for a multitude of reasons. Specifically, optogenetic tools allow for greater specificity through avoidance of secondary chemical effects. Availability of multiple isolated wavelengths allows for easy combinatorial implementations. Modern laser microscopy enables precise 3D localization of effect, and physical barriers can often be overcome through the use of light rather than chemical compounds. These tools can often be adjusted within milliseconds, allowing for specific cellular manipulation, and can be used to study a broad range of cell types in live animals, especially during development when tissue depth is minimal and tissue transparency is greatest. Initiation of proteins by exposure to specific wavelengths of light is a straightforward and reliable mechanism to modulate protein function in vivo. Degrons are naturally occurring peptide markers which facilitate degradation of the protein to which they are attached. To be used as optogenetic tools, these degradation tags are fused to cryptochromes, enzymes which change conformation in response to light to expose or conceal cryptic domains within themselves, allowing for concealment and longevity of the target protein or exposure and degradation of the target protein in response to light. While optogenetic degrons as a class are often referred to as photosensitive degrons, PSD is a specific optogenetic degron developed by fusing the photosensitive domain of Arabidopsis thaliana, phototropin 1, light oxygen voltage 2, LOVE2, to mouse ornithine decarboxylase carboxy terminal degron, so CODC. When fused to a protein of interest, exposure to blue light induces a conformational change to expose the cryptic degron, leading to protein degradation of the construct, which can then be repopulated in the dark. Another system developed for triggering protease degradation of fused proteins by exposure to light is the blue light inducible degron, BLID method. BLID contains a small peptide degron rendered cryptic by fusion to LOVE2 domain. In this way, degradation of fusion proteins is triggered by exposure of the degron upon exposure to blue light. Photo N degron was recently developed to take advantage of light-dependent end rule mediated protein degradation. Light-induced uncoiling of the J-alpha helix in LOVE2 exposes an N-terminal arginine amino acid and triggers end rule degradation. Apart from degrons, there are other categories for deactivation of proteins. One such method is photosensitizer, used in chromophore-assisted light inactivation, CALI. Photosensitizers are chromophores which produce reactive oxygen species in response to light activation. CALI takes advantage of this production to inactivate proteins attached to a photosensitizer. The first genetically encoded photosensitizer was killer red, derived from the hydrozoan chromoprotein ANM2CP, which produces phototoxic effects via reactive oxygen species production in response to green light exposure. Killer red is an effective optogenetic tool for selectively killing cells and tissues through green light-mediated reactive oxygen species production, but is also a useful tool in CALI schemes. Because CALI is a more direct system than degrons, inactivation is substantially more rapid than degron-based systems, but have the drawback of producing cytotoxic reactive oxygen species. Since killer red, multiple alternative photosensitizers have been developed to counter killer red's propensity to dimerize and facilitate use of their wavelengths of light. Lights Out introduced an ASLOVE2 domain into the GAL4 transcription factor, widely used for conditional transgenic expression, between DNA binding and gene activation domains to suppress GAL4-mediated expression when exposed to blue light. Next subsection, subcellular translocation systems. ASLOVE2 has further been implemented in light-activated nuclear shuttle, LANS, and light-inducible nuclear localization signal, LINUS, to enable conditional nuclear localization of proteins of interest upon exposure to blue light, using a cryptic NLS signal. Conversely, the light-inducible nuclear export system, LINX, and LEXI employ a light-exposable cryptic nuclear export signal within an ASLOV fusion, allowing for blue light-induced nuclear export of proteins of interest. The ability to reversibly trigger BCLOVE4 membrane binding blue light exposure has enabled membrane binding via blue light exposure, has enabled its use as a conditional fusion protein localization system, whereby blue light is used to target fusion proteins to the membrane. An interesting naturally occurring optogenetic tool for localization to DNA is EL222, which dimerizes and binds to DNA in response to blue light. This system is currently used in multiple contexts to control transcription via light exposure. Next subsection, binding and polymerization systems. An enormously useful optogenetic innovation has been transgenic tools which facilitate manipulation of binding dynamics of proteins of interest. It is now possible to use these tools to hold proteins in contact with one another, sequester them against membranes, or release them via exposure to laser light. This mechanism is not only of inherent utility in protein of interest functional control, but also has been shown extensively to be greatly advantageous as a compounding factor in conjunction with other optogenetic mechanisms. ILID, an improved and modified version of LID, is a blue light inducible dimerizing protein which forms a dimer with SSPB in light and dissociates in the dark. ILID utilizes an ASLEV2 domain fused to an SSRA peptide, which is then able to bind its binding partner SSPP upon exposure via reversible light-inducible LOVE2 conformational transformation. A deservedly popular use of the eyelid system is OptoSOS. OptoSOS, pardon me, I lost my place again. Is OptoSOS. OptoSOS utilizes a membrane-anchored eyelid to recruit SSPB fused SOS to trigger a RAS-ERK cascade when exposed to blue light. Another light-triggered dimerization system is cryptochrome 2, Cry2, and CIB1. In this system, blue light induces heterodimerization of Arabidopsis thaliana, Cry2, and CIB1. Simultaneously, however, the system also causes Cry2 homooligomerization, creating both a potential experimental challenge and a useful mechanistic expansion beyond other heterohomodimerization systems. Intriguingly, an optogenetic heterodimerization system exists which utilizes UVB light. In the presence of UVB, Arabidopsis COP1 binds to UVR8 and can be used to bring proteins of interest into proximity to one another in living cells. Similarly, heterodimerization can be controlled by red and far red light, as in the phytochrome B, phytochrome interacting factor PIF system. However, these red or far red phytochrome systems require plant chromophore 3Z PCB, which is not naturally synthesized in most animals, making them challenging to use in transgenic systems. Nevertheless, PCB can be synthesized in mammalian cells by transgenically introducing 4 genes required to generate it from heme. An intriguing use of the system is SOS-CAT, or SOS-CAT, which operates identically to eyelid OptoSOS system via red and far red light rather than blue, allowing for a greater freedom in designing combinatorial transgenic systems. Furthermore, Arabidopsis thaliana, Phi-A, FHY1 behaves similarly, allowing for the creation of RedMap, which facilitates gene activation under red light through heterodimerization and deactivation under far red light via dissociation. Bacteriophytochrome BPHP1 is used as a light-sensitive binding partner to PPSR2. More recently, a truncated version of PPSR2, QPAS1, was engineered to reduce its size and mitigate natural oligomerization of PR2. In this light-sensitive binding method, far red light exposure causes binding of BPHP1 to its binding partner, which is reversed in red light or darkness conditions. A further innovation in light-dependent interaction, magnets has been developed to induce protein of interest heterodimerization in the presence of blue light. Magnets was developed by modifying the pass domain of the Neurospora crassa love protein vivid, which forms heterodimers when exposed to blue light. Modifications were made to the protein sequence in the binding domain to either introduce positive PMAG or negative NMAG amino acids, thereby creating 2 modified vivid photoreceptors which heterodimerize but do not homodimerize in the presence of blue light. Another tool derived from vivid is light on, which uses Gal4 fused to vivid and a P65 activation domain, GAVP, to induce dimerization and activation of UAS target gene expression when exposed to blue light. Contrary to the bind in the light, dissociate in the dark scheme of the systems discussed so far, there also exist systems which employ the contrary scheme. An example of this is PIXD-PIXE system. In the dark, PIXE and PIXD form oligomers containing 5 PIXE and 10 PIXD proteins, which dissociate into PIXE monomers and PIXD homodimers when exposed to blue light. Interestingly, bacterial photoactivated adenylyl cyclase, BPAC, which utilizes a BLUF light sensor domain similar to PIXD, facilitates light-dependent increase in C-amp levels, in cyclic amp levels, in LOVE2 trap and release of protein, so LOVE trap. A small protein, Z-dark, ZDK, was developed to bind to the dark state of LOVE2. This facilitates the conditional tethering and release of LOVE2 to ZDK under dark or blue light exposure, respectively. Somewhat orthogonally paralogous to the ZDK LOVE2 binding pair is the arisen PDZ LOVE2 binding pair, tulips, whereby a CPE PDZ can bind to a short peptide sequence fused to LOVE2 upon blue light exposure. A disparate mechanism of light-mediated anchoring control is through light-controlled protein cleavage. An example of this system is photocleavable. Photocleavable consists of a modified circularly permutated M-maple, which spontaneously dissociates upon exposure to violet light and disconnection of N and C terminally fused proteins of interest. The next subsection is opsins. Perhaps the simplest example of optogenetic tools are the opsins, which have gained immense popularity in neural studies and are lurching toward translational implementation. Opsins are light-sensitive membrane proteins which regulate transmembrane ion transfer. This makes them simple, inherent optogenetic tools whereby membrane potential can be controlled by light exposure. An example of an opsin optogenetic tool is Phoenix, an optical proton pump tool. So Phoenix uses a modified ARCH3 light-sensitive proton pump fused to an ATPase, vesicular protein synaptophysin, and optical pH sensor fluoron to facilitate proton shuttling to acidify the interior of synaptic vesicles via yellow light and pH detection via blue light. Similarly, several other optogenetic opsin tools are available which facilitate light-mediated transmembrane proton pumping, sodium ion pumping, and chloride ion pumping. Furthermore, guanylyl and adenylyl cyclase rhodopsins, such as rhodopsin guanylyl cyclase, rhodopsin cyclic nucleotide phosphodiesterase, and guanylyl cyclase rhodopsin, have demonstrated utility in mediating light-dependent cyclic AMP or cyclic GMP levels to exogenously regulate subcellular signaling. That brings us to Section 4, Modern Advances in Combinatorial Real-Time Optical Methods. Techniques involving simple, direct, light-dependent messenger manipulations are quite popular and elegant. However, the most exciting implementation of transgenic optical tools are those which involve cooperative implementation of multiple systems simultaneously to enable greater control over the comprehension of the molecular biological systems being interrogated. It is feasible that experimental protocols can already be developed to visualize, measure, and manipulate one or more subcellular molecular processes in the same cells, in real time or near real time. Indeed, a growing number of such systems combining multiple transgenic optical tools and strategies are being developed to combine or improve upon individual schemes. A complex blend of transgenic optical tools, the blue light inducible TEV protease BLITZ system, combines Cry2 CIB1 system with the ASLOV2 system to precisely induce transcription upon exposure to blue light. BLITZ consists of two parts: a transmembrane bound fusion of CIBN, a truncated version of CIB1, the N-terminal portion of the tobacco H virus protease TEV, ASLOV2, TEV cleavage site, and a transcriptional activator, and a soluble portion consisting of Cry2 PHR, a truncated version of Cry2, and the C-terminus of TEV, CTEV. Upon exposure to blue light, a binding of Cry2 PHR to CIBN facilitates interaction of the two parts of TEV while simultaneously exposing the TEV cleavage site, allowing cleavage and release of the transcriptional activator. Another tool in the modern genetic engineer's toolbox is the light-activated reversible inhibition by assembly trap, Lariat strategy. Lariat consists of a CIB1 bound CA-MK2 alpha and a Cry2 bound protein of interest. Since CA-MK2 alpha self-oligomerizes, exposure to blue light reversibly traps the protein of interest in clusters. The PIX-E-PIX-D heterodimer scheme has also been used in to conditionally induce and dissociate subcellular protein droplets in the pixels system. In pixels, blue light exposure diffuses phase-separated droplets formed in the dark through a combination of liquid-liquid phase separation function of N-terminal intrinsically disordered protein regions, IDR, of the human FUS protein, FUS-N, and the oligomerization of PIX-E, PIX-D under dark conditions. A similar mechanism to pixels developed in the same study is optodroplets, which uses Cry2. In optodroplets, FUS-N is fused to Cry2 to form phase-separated droplets when blue light is applied to promote oligomerization of Cry2. Lands trap and clasp both improve upon the Lands system for LOV2-mediated nuclear import. Lands trap uses a membrane-bound ZDK2 to directly bind Lands in the dark, whereas clasp uses a membrane-bound LOV2 to trap ZDK protein of interest Lands fusion in the dark, thereby improving nuclear exclusion of Lands pre-blue light exposure. On the other hand, Links Nano fuses links to nano, a truncated version of SSPB, which facilitates binding to a membrane-bound eyelid in blue light. This improvement of the link system produces more complete nuclear export by tethering exported links nano protein of interest fusions within the cytoplasm. Similar to Lands trap and clasp, in function is iris. which sequesters the LOVE2 NLS fused protein of interest in the cytoplasm via fused QPAS1, which facilitates binding to a membrane-anchored BPHP1 when exposed to far red light, and nuclear import by NLS exposure when exposed to blue light. Specific protein-associated tool giving transcriptional readout with rapid kinetics, SPARC, is a tool devised to conditionally report cells in which two proteins of interest are interacting. One protein of interest is fused to a TEV protease, while the other is fused to a modified LOVE2 TEV cleavage site, fused to a transcription factor. Upon exposure to blue light, the TEV cleavage site is exposed, allowing for release of the transcription factor and activation of the reporter genes in cells where the two proteins of interest are interacting. Bacteriochrome-based light-inducible dimers, Bicycle, employs a modified induced isomerization of GAF domain derived from Caryochlorus marina, AMG2, to shift binding between two binding partners when exposed to either red or green light. Binding partners, binder of AMG2 red state, so BAM red, and binder of AMG2 green state, binder of AMG2 green state, BAM green, were engineered such that AMG2 binds BAM red when exposed to red light and to BAM green when exposed to green light, allowing for protein of interest swapping via light exposure. The developers of Bicycle showed that it can be used to conditionally recruit either a transcriptional repressor or activator to DNA-tethered AMG2, depending on laser color choice. Blue off combines LOVE2-mediated transcriptional inhibition with PSD-mediated protein of interest degradation to more completely eliminate protein of interest presence when exposed to blue light. LOVE2 is used to create a cryptic DNA binding domain and is further fused to a transcriptional inhibitor, CRAB, to eliminate transcription of the protein of interest, which is fused to BLID for degron-mediated depletion. Analogously, iLight controls gene transcription via far red-mediated homodimerization of photosensory core module ideomarina, ISPAD-C, or ISPAD-C PMC, fused to either LEX-A408 to block transcription or GAL4 and VP16 to trigger gene expression, which can be reversed via exposure to near-infrared light. In the dark or after exposure to near-infrared light, the fused transcription factors are sterically inhibited by isomeric transformation of ISPAD-C PMC. All right, that brings us to the discussion. While there have been many reviews on optical tagging systems, optical biosensors, optogenetics, and biologically expressed imaging tools more broadly, we have focused this compendium specifically on tools currently available for use in transgenic plant and animal studies. In a few decades, molecular biology and genetic engineering fields have exploded from complex transgenics being a hypothetical potentiality to thousands of transgenic organisms, hundreds of fluorescent proteins and tags, scores of subcellular sensors, and dozens of applications for several optogenetic systems. It is important to carefully consider the limitations of each class of optical tool as well as for each individual instrument therein. For example, labeling with one of the tag systems described here comes with the disadvantage of occupying a laser and detector on the microscope, whereby to label. So, it's sometimes prudent to use a sensor also as a label, which may come with the disadvantage of slow fluorescence maturation and low signal. Similarly, within classes of optical tools, different tools with similar functions have important advantages and limitations to consider. For example, degrons require transcription and translation of replacement fusion proteins of interest to recover after depletion, while translocation tools require mere shuttling within the cell to recover. However, degrons more quickly and completely deplete protein of interest levels. Further, it's necessary to contemplate the activation-inactivation times of optogenetic tools, which often take a couple to tens of minutes to reach peak activation, as well as their efficiencies of activity and reversibility, which tend to land all over the map. In addition, when considering the use of sensors or tags in combination with optogenetic tools, it's imperative to weigh the cost in photobleaching and phototoxicity against the reward of maintained optogenetic activation and tailor experiments accordingly. As discussed in the previous section, combinatorial applications of these tools are being developed which synergistically supplement each other. Each individual complements limitations. It is expected that we will soon see a surge in development of sophisticated tools and studies monitoring chromatin architecture, transcription, translation, protein localization, and cellular responses via reporters, tags, and sensors, while simultaneously manipulating transcription or protein levels or functions in living transgenic plants and animals. One such study has already reached preprint from the lab of Hernan Garcia, manipulating transcription factors via Lexi while simultaneously monitoring transcription of their targets via MS2 MCP. Another ongoing study from the lab of Sanjeevi Sivasankar seeks to combine optogenetics and bioID by fusing Cry2 or CIB1 to two halves of the split turboID biotinylating enzyme, thereby allowing for precise temporal control of the enzyme's activity. Additionally, we have conceived a few hypothetical advancements which we believe can be useful if developed. First, precise spatial control of epigenetic states has the potential to be an invaluable tool for interrogating development and disease, and can feasibly be accomplished by ASLEV2 toggling of histone modifiers and genome targeting via gRNAs and dCas9. Second, combining tags with other systems is sure to soon gain popularity. Using SunTag to sequester transcription factors conditioned upon precellularization conditions in the early Drosophila embryo, for example, could be useful for specific inquiries into early development. Third, the Blitz system can feasibly be modified to control overexpression transgenic transcription factors, which would otherwise accumulate in systems like Lynx Nano and confound expectations of spatiotemporal precision. Nanotoxic blue and red light are certainly most useful for in vivo studies in transgenic animals. Complex systems non-toxic blue light. I thought that was wrong. Apologies. Non-toxic blue light and red light are certainly most useful for in vivo studies in transgenic animals. Complex systems incorporating varieties of optogenetic tools will require multiple lasers to be used in concert. It is exciting that several systems have already been developed which implement Bohemian wavelengths for activation, including far red, near infrared, violet, and UVB. With appropriate experimental design and controls, it is hypothetically possible to simultaneously use a handful of different lasers to affect different optical tools and achieve increasingly complex objectives. We will undoubtedly see further exponential growth of the field of transgenic optical tool development and application in the coming years. Neurological and metabolic disorders stand to benefit greatly from future advances, which will undoubtedly include translational applications with medical potential. Encouragingly, advances in ex utero and in utero experimental techniques and technologies promise to bring optogenetics to mammalian embryonic development, an important step towards this goal. Further, the genetically encoded devices described herein offer our best chance at supplementing our insufficient understanding of basic cell developmental and disease mechanisms, which have hitherto remained inaccessible. This review is meant to be a brief almanac of the tools available to those who will develop and use these future models and devices. And that's effectively the end. Thank you for listening, and I'll see you next week.